Hello everyone. In this video I'm going to be talking to you about enzymes, structure and function. This is suitable for year 9, 10 and 11. Most students do enjoy learning about enzymes, but sometimes in exam questions they miss out key details that are required for the higher grades 7 to 9. Enzyme structure. Enzymes are made of proteins. They have a 3D shape and they have a specific region called an active site. Often you might have seen enzymes being drawn looking like Pac-Man. That is my enzyme. This part here is the active site. The function of an enzyme. Enzymes are biological catalysts. This means they speed up the rate of chemical reactions in living organisms. The enzymes are commonly involved in digestion reactions. If you have already learned about digestive system, you may remember an enzyme called amylase that's found in the mouth, and that amylase digests starch. Enzyme catalyzes a reaction when the substrate binds to the active site. The substrate has to be a specific shape that will match the active site. The key word we use is complementary. The substrate is complementary to the active site. Once the substrate joins to the active site, the name for this structure is an enzyme substrate complex. ESC for short. The enzyme will then break the bonds in the substrate so that products are formed. The enzyme remains unchanged. The enzyme is not used up. So the enzyme can catalyze more reactions. The mechanism for the way in which enzymes work is called the lock and key theory. The enzyme is the lock. The substrate is the key, because only the correct key can be used to unlock the lock. For this reason, we say that enzymes are very specific. One enzyme catalyzes the reaction for one substrate. Some examples. I already mentioned one enzyme that you may have heard of that's found in your mouth. This is called amylase. Amylase enzyme digests starch, which is a large polysaccharide, into a disaccharide, called maltose. Another enzyme, protease, catalyses the reaction to digest protein into amino acids. Lipase enzyme catalyses the reaction to digest lipids into fatty acids and glycerol. Enzymes are affected by different conditions. Temperature and pH have to be correct for enzymes to work the best. The temperature at which enzymes work the best is called the optimum temperature. In a human, the optimum temperature would be 37 degrees because that is the body temperature of a human. However, in a plant, the optimum temperature would be lower, maybe 22 degrees. So not every organism has the same optimum temperature. If the temperature is very low, the enzyme, ENZ for short, and the substrate, SUB for short, do not have much kinetic energy. You would have heard of this word in physics and also possibly in chemistry. If the enzyme and substrate do not have much kinetic energy, there will be fewer successful collisions between the substrate and the enzyme's active site. This means there will be fewer ESCs. Can you remember what that abbreviation was? Enzyme substrate complexes, resulting in a slower rate of reaction. So a low temperature leads to a slower rate of reaction. But if the temperature is high, if the temperature is above the optimum, the enzyme vibrates too much. Bonds in the enzyme start to break, and the active site changes shape. This is a problem because the active site is a very specific shape so that the substrate can bind. But if the active site changes shape, the substrate cannot bind. We say that the enzyme is denatured. This is a very important key word. The enzyme denatures. This is not saying the enzyme dies. An enzyme cannot die because it is not alive. However, the enzyme denatures. Therefore, there's going to be a slower rate of reaction. We did not use this word denature when talking about low temperatures, only when temperatures are high above the optimum. Another variable which will affect the rate of enzyme-controlled reactions is pH. The pH at which enzymes work the best is called the optimum pH. For example, in the mouth, pH 7 is the pH inside of the mouth, so therefore enzymes in the mouth, like amylase, will have an optimum of pH 7. But different parts of the body have different pH. The stomach is very acidic due to hydrochloric acid. So this means the enzymes have a much lower pH. for their optimum, for example, pH 2. If the pH is above the optimum or below the optimum, we can use this keyword denature again because the active site changes shape. So that same keyword, the enzyme denatures. This means the substrate cannot bind to the active site and there'll be fewer enzyme substrate complexes. And a slower rate of reaction. So in this video, we have discussed the structure of enzymes, the function of enzymes, examples of some enzymes and the reactions they catalyse, how enzymes are affected by temperature, and how enzymes are affected by pH. I hope you found this video useful. If you like this video, please give me a thumbs up. Please add a comment and definitely subscribe. If you'd like to have videos about any other topics, let me know in the comment section. Bye.